So good morning everybody, I am Soumyabrata Dey and thanks for coming to my dissertation presentation, which is about automatic detection of brain functional disorder using imaging data. And the particular disorder we are working on is called attention deficit hyperactive disorder, or in short ADHD. So the problem definition is pretty easy. Given the brain imaging data of a subject, is it possible to design a system which can automatically classify if the subject is affected by ADHD or not? And we used for this purpose two data modalities. It's called functional magnetic resonance imaging data, or in short fMRI, and structural magnetic resonance imaging data, or in short SMRI. So before going into the problem details, I want to talk a little bit about the background of ADHD, the motivation of our work, and also a little overview of the different data modalities we used. So first, ADHD background. So ADHD is most commonly found childhood brain disorder, and it's found out like 5 to 10% of the children all over the world is affected by this problem, which is a pretty huge number. And the main symptoms are inattention, hyperactivity, and impulsivity. And so sometimes the subject cannot concentrate on one task. Sometimes they find it difficult to follow school education. So this is kind of a day-to-day problem for them. Also, the root cause of this problem is not clear, which makes the doctor to go into a manual diagnosis procedure where subjects are put into a controlled environment and interviewed there, and based on the interview they are detected as ADHD or not. So this kind of diagnosis method has some problem. First, many of the time, like general physicians perform this diagnosis who actually don't have the required expertise for this kind of job. And it's found out like 20% of US children by the time they turn 18 are diagnosed with ADHD, and this 1:5 ratio is a pretty alarming number, and many experts believes like this is a clear sign of overdiagnosis of the problem. Also, this method suffers from lack of standards due to the human judgmental error. So these are the issues which motivate us to go into design a automatic diagnosis of ADHD system, which we expect to reduce these problems. So though the root cause of the ADHD is not yet clear, there are some hypotheses behind the ADHD. So one of them is like a lack of neurotransmitter like dopamine in our brain. So the function of this neurotransmitter is to make communication between different regions of the brain to perform some cognitive task. And lack of presence of this neurotransmitter sometimes can hamper the communication network. Second, is found like statistical difference between the functional activity of the ADHD and control group of subjects, and also researchers found different abnormal growth in different brain regions in the ADHD subjects. So one of the key regions of for ADHD is called resting state network, or RSN, and the name is given because this is the region which remain active when we don't even perform any conscious task, or even when we are sleeping. So one of the part of this region is called default mode network. It's part of the RSN, as I said, and its task is daydreaming and introspecting. And many actually functional differences actually found in default mode network, and it's kind of a key area for studying ADHD. So based on this finding, we actually model our system where we model the brain functioning as a connectivity network, and we try to exploit the topological difference between ADHD and control subject in our classification method. So let's look into the data. So first, the fMRI data. So it's found out like different brain regions perform different tasks, and when they perform tasks, they require more energy, and this more energy is supplied by the blood oxygen level. So fMRI is a technique which can detect the blood oxygen level, and the brighter intensity in the fMRI data is actually related to higher level of oxygen in the brain, which indicates like higher level of activity in that region. So fMRI can be assumed as a 3D brain video which captured the brain activity dynamics, and each frame of this video is a 3D frame where two consecutive frames are captured approximately 3 seconds apart. So the smallest unit of fMRI data is called voxels, which are small cubical regions. So here we are actually showing a real fMRI data. In the first three images, we are showing three cross-segmentation view in three orthogonal directions of the brain, and the red cross is showing a particular voxels in the three view. And the fourth quarter of the image, we are plotting the fMRI time series for that particular voxels. So fMRI data can also be assumed as a collection of these fMRI time series for all the voxels of the brain volume. On the contrary, structural MRI data is a static 3D brain image, but the resolution of SMRI data is pretty high, typically 4 times higher than the functional MRI data. And here is the example of SMRI data where you can see we can view different tissue structure of the brain in SMRI data. So now we want to give the details of our method. We started with a simple approach called bag-of-word framework, and then gradually we actually improve our model, taking care of the shortcomings of the previous approach. So this work is published in a medical imaging conference called SPI. So the main approach is, first we model the brain function as a functional connectivity network where different regions of the brain are represented by the node of the network, and those nodes are connected by some edges if the activity pattern of those regions are pretty high. Activity patterns similarity of those regions are pretty high. Then for each node of the network, we compute degree feature, and we represent each network as a histogram of degree feature using the bag-of-word framework. Finally, we use SVM for the final classification. So before going into the method details, I want to provide some information behind the bag-of-word approach. So this approach is actually originated in document classification. What it says, given a document, the document can be classified only by the occurrences of different words in the document. So given a document like this and a dictionary which contains all the words possible, we can count each word's occurrence in the document. For example, Brazil occurred twice, World Cup occurs thrice. Similarly, we can compute the word occurrence for all other words to represent the document as a histogram of words. So now, given a set of training documents, we represent each document as a histogram of words, and we train a classifier based on this histogram and using this histogram as a feature vector. Then given a test document, we represent it again as a histogram and use the train classifier for the final classification. So this kind of approach is the main problem of adopting the bag-of-word approach in image or video classification is the concept of visual words, which is not very obvious. So what we do, given a training set of images, we extract local spatial features and project those in a actually feature space. And this feature space can be anything depending on your framework. And then we cluster these features on the feature space, and each of these clusters are then represented is a representation of a visual words. So then given a test image, we again capture these local features and project them in the feature space and assign them to the nearest cluster so that we can represent any image by the histogram of visual words. So rest of the document classification framework is similar to the rest of the classification framework is similar to the document classification framework as I described. So given this background, let's describe what is our method. So given a 4D fMRI data, we first arrange it as a matrix where each column of the matrix is a fMRI data time series for a voxel, and all the voxels are arranged in a row. Then we compute the correlation matrix whose size is n by n, where n is the number of voxels in the brain volume, where each cell of the correlation matrix compute the similarities of two fMRI time series of two voxels. Then we can use some threshold to binarize these correlation values to get a adjacency matrix which can represent a network. So a 1 in particular cell of ith row, jth column signifies that ith and jth voxels activity patterns are very similar and they are connected by an edge. So after representing as a network, we compute like network feature, which is degree for each node of the network, and we represent each node as a 4D tuple where D is the degree of the network and XYZ are the physical coordinate. Then using the Bagotvert framework, we clustered them in the four-dimensional feature space to represent each subject as a histogram of these 4D tuple features. And we use 100-dimensional histogram here. Finally, we use SVM as the final classifier. So correlation of two time series U and V can be computed by a simple formula like this. This is a pretty common formula. It's called Pearson correlation formula. So the whole Bagotvert framework is like this. Given n subject, we first represent them as networks and then compute the degree features for each subject to represent each node as a 4D tuple, and then use the Bagotvert framework using k-mean clustering to cluster them to form the code book, and then perform the feature quantization to represent each subject as a histogram. For comparison, we used a different framework where each subject is represented by the original time series, because we actually represent each voxels of the brain volume by its original time series. And then we perform again the same framework, k-mean clustering, generation of different code book using feature quantization, and then we can represent each subject by a different histogram. So given a test subject, we can extract these two features, and using two different code book, we can quantize them into like degree histogram and raw time series histogram. And third representation we used is the concatenation of the degree and time series histogram. So for the experiment, the dataset we used is called ADHD 200 dataset. It's released for a global competition for classifying ADHD using some classifier, and also to understand the pathopsychology of the problem. This dataset was released. It's a pretty huge dataset. It contains information from 8 different data centers. And first 776 subjects were released for training the classifier. Later 197 subjects were released to validate the classifier performance. It's called holdout set. So actually 51 team registered for this competition, and using our approach we got like 12th position. So ADHD 200 dataset contains 3 different modalities of data. First, structural image data. Second, the functional videos, and finally the phenotypic information, which contain age, gender, handedness, IQ, etc. So here is a snapshot of the dataset. The top row is actually showing different data centers for release set, and bottom rows are showing the data centers for holdout sets. For experimental setup, for histogram, we use 3 different kind of histogram as stated. We use degree histogram, which is 100-dimensional, time series histogram, which is 75-dimensional, and this dimension are selected empirically. And finally, we use a combined histogram, which concatenate 2 histogram of, and dimension is 175. For classifier, we use SVM, and we use 2 different classification setup. In the first setup, we use 506 subjects from the release set and perform leave one out cross validation. In second setup, we train our classifier using the release set subject and verify the classifier using the holdout set subjects. So the first classifications results are like these. Using degree feature, we got 61%. Using raw intensity time series, we got 56%. Using the combined feature, we got 64%. For the second kind of experimental setup, we got kind of similar kind of results, like 62% using degree map, 56% using intensity time series, and around 65% using the combination of the both feature. So the main contribution of this work is first is one of the few initial attempts for automatic classification of the ADHD subjects, where we model the brain as a functional network and exploited the topological difference of the ADHD and control subjects. And there are actually few shortcomings of this method. First, we lost some spatial information while constructing the histogram, because histogram only counts the occurrence of different features. And second, we use actually features from the whole network, but sometimes some part of the network might not be active because in the resting state all brain is not active. So including data or features from those regions might not be useful. Finally, we only analyze the degree feature, but there can be other network feature which can be useful for the classification of the ADHD. So to overcome these problems, we move to the next, our next approach, which is network feature analysis. And this work is published in Frontiers Neuroscience Journal. And so the main improvement performed by this approach is, first, we proposed an novel algorithm for identifying useful regions of the brain which we believe is useful for ADHD classification, and we experimentally showed like selecting features from only those regions actually improve the classification accuracy. Secondly, we analyzed different network features such as degree map, K-cycle map, and weight map. Also, we form the feature vector by concatenating the feature of different nodes of the network in certain order to preserve the relative spatial orientations. So the framework until the network computation stage is pretty much similar to bag-of-word framework. After computing the network, we can extract different network features from the networks and then use our algorithm for useful region mask collection to identify those regions important for ADHD classification, and we form our feature vector from only those regions, the features from only those regions, and finally we use PCLDA classifier for final classification. So here are the different features we use. First, degree. So degree of a node is the number of edge it is connected to. For this example, it's 5. Similarly, degree can be computed for other nodes to form the degree map. For 3 cycle, we compute like how many 3 length cycle a particular node is participated in. For this example, this node it participated in 1, 2, and 3 cycle. So its 3 cycle count is 3. Similarly, 3 cycle count can be computed for other nodes. So varying distance degree map not only compute the degree of a node but also the distribution of the edge length. For example, here this node is connected to 2 short range edge, 2 mid range edge, and 1 long range edge. So its degree is a small vector. In this particular example, it's 2 to 1. For weight map, we don't consider each node as a binary value, but we actually consider its actual correlation value. So we sum up all these correlation value for a particular node, and for this example its weight is 3.65. Similarly, the weight can be computed for other nodes too. So in this slide we explain our useful region mask computation algorithm. Though the algorithm is computed on like the 3D brain regions, but for the easiness of representation, we presented it on a 2D slice of the brain. So given the brain volume, and first we subdivide the brain volume into small cubical regions, and we then select a subset of those regions, and we extract feature only from these regions to form the feature vector. Then we perform classification using those feature vector and report the accuracy. So this subset selection is performed many a time, each time extracting feature, classifying, and reporting results. So given this idea, the whole algorithm is like this. Given the subdivisions of the brain volume for all the subjects, first we actually select different subset of the regions, and for each of these subset we perform the classification and reported the accuracy score. Then we rank this subset based on their classification accuracy, and we select only the top 10% of the subset. Then we actually compute the occurrence of each region in the top performing subset, and then we normalize those values between 0 to 1. Then if we can use a threshold on those probability map, we can extract the most useful regions which occurred most frequently in the top performing subsets. So here are the most useful regions map we found out, and it's plotted on the structural image of the brain. And we also showed like these regions are independently found by other researchers who worked on ADHD classification or ADHD significance on the brain. So we used two experimental setups. In the first setup, we used only KKI release set, which counted 83 subjects. We used first 44 subjects for training, last 39 subjects for testing. And for the features, we used degree map, variable distance degree map, K-cycle map, and weight map. And finally, we used PCLD classifier. So here is the result for the first setup. So two main things to indicate here. First, we got best classification accuracy is in almost all the cases when we are using correlation threshold at 0.80. So this value is used for future experiments. Secondly, when you are using the useful region mask to extract feature, we are always getting better performance compared to when we are not using. For the second experimental setup, we used release set subjects for training and holdout set subjects for testing. For feature, we validate two kind of feature, degree and 3-cycle map. For the classification, we use PCLD classifier. So we use actually two different parameter also, which is called sensitivity, which actually true positive rate, which indicates like number of true positive identification compared to the all positive samples present in the dataset. Similarly, specificity indicate true negative rate, which is number of true negative classification compared to like all the negative samples available in the dataset. So here is the results for all the data centers, and as I said, we computed the performance of two features, degree and 3-cycle map, and we got around 70%, which is the best classification accuracy we got using this method, using 3-cycle map feature, though both of the features perform equally well. And also we showed like when you are not using the useful region mask, we got a lower performance compared to when we are using it, and we got around 5% improvement when you are using the useful region mask. So the main contribution here, we performed, actually we compute the performance of different network features in this analysis, and we also propose a useful region selection algorithm, and which help us to reduce the data dimension, improve the classification accuracy. Also those regions are independently found by the other researchers. Also the detection rate was one of the top performing at the time of the publication. So few shortcomings of this method is first, we're actually capturing the local node based feature on the network, which can capture the local network structure, but it actually ignores the global information of the network. Secondly, we represent each voxels of the brain as a node of the network, which makes the network computation cost pretty high. So keeping in mind these shortcomings, go into our next approach, which is based on attributed graph representation. So this work is published in Frontiers in Neural Circuit in June 2014. The main motivation of this work is first, we try to incorporate the global topology of the networks in our classification framework. Second is we try to efficiently represent the network, keeping like most relevant information with minimum redundancy such a way that the network computation cost is reduced. So the main approach is we represent a cluster of voxels as a node of the network, and each node is represented by an attribute set, and based on this attribute set we compute the inter-network distance. And after computation of inter-network distance matrix, we use that for project the network in a lower dimensional space where the final classification is performed. Such a way we make sure we use the global structure of the network. So here is the framework of our approach. Given the 4D fMRI data, we actually compute 3D power map image where power for each voxel signifies the activity level of that voxel, and I'll come into details how we compute the voxel power map. So then we actually select the high power voxels, and we perform clustering on those high power voxels, and each of this cluster is represented as a node of the network, and two nodes of the network is connected by an edge if their activity patterns is pretty similar. After computation of the network for each of the subject, we compute the network to network distance, and after computing all pair of networks distance, we form the inter-network distance matrix. And this distance matrix is used to project the network in a lower dimensional space using the multi-dimensional scaling technique. And the final classification using SVM is performed there. So here is the formula for computing a power map given a given time series t. The power of that time is the summation of the square term and averaging that out. So the power map is formed by computation of average power for each voxel of the brain volume. And what is the significance of the power map? First, it can select the highly active voxels, and we believe like highly active voxels are more informative, and lowly active voxels which is not performing any task during the resting state don't contain any useful information for classification. Second is it helps us to reduce the data dimension. Also, it helps us to reduce the noise in the system, because if you incorporate those lowly active voxels in the network construction step, it might negatively affect the classification accuracy. So the functional connectivity network is computed like this. First we select like high power voxels of the brain volume. Then we cluster those voxels over the space using a region of interest mask is called CC200 map. And this map looks like this. It actually segments the brain volume into 190 regions, and those regions are spatially contiguous and functionally homogeneous. So the highly active voxels of each of those regions are clustered together to form the nodes, and then network is constructed by connecting any pair of nodes whose average fMRI time series correlation values is higher than some threshold. So then each node of the network is represented by attribute set. For example, a node is represented by its degree, its average power, its degree of the neighboring node, average power of the neighboring node, and the spatial position of the node. So each node is actually represented by a small vector A which contains 5 attributes. So then we compute the intergraph distance matrix, and it's a two-step process. First we compute the node-to-node distance. So given two node A and B for two networks like this, we compute the distance between two nodes using this formula where W is the weight vector. So after computing the distance for each pair of the nodes of the two networks, we compute the node-to-node distance matrix, and this distance matrix is used for graph pair distance computation. So given this node-to-node distance matrix, we use Munkar algorithm to assign each node of the first network to one node of the second network such a way that the total assignment cost is minimized. And this final matching cost is represented by the graph distance between two graphs. So we compute this graph distance for all pair of the networks we computed to come up with the intergraph distance matrix. So the main idea of graph distance is given a network like this and another network like this. So spatially closed nodes should match given their local structure is similar. For example, these two nodes are spatially close, but their local structure is not as similar as these two nodes. So these two nodes should match. So after this, we actually project the network in a lower dimensional space using MDS technique. So given the distance matrix DG, MDAs project the graphs to a lower dimensional space such that this formula is minimized, where DG dash is the distance matrix in the projected space. So advantage of MDS is this method works even when the original space is unknown, which is true in our case because we don't know what space these networks belong to. So the experiments we perform first tenfold cross validation on the release set. Second, we trained our classifier using the release set subject and perform the validation using the holdout set. And for the final classification, we use two approach. First, we perform the classification on the all subjects. Second, we perform classification separately on male and female subject group. And we use SVM for the classifier. So here is the result on the whole data set. As I said, we perform classification on the release set, which is on the left side, and holdout set, which is on the right side. Also, we showed when you are performing classification on the all subjects, the accuracy is lower compared to when we are classifying on the male and female subjects separately. So the possible explanation for this is it's found out the activity pattern of the brain in the male and female subject is pretty different. So if we classify it separately, it's probably helping the classifier to better understand or identify the ADHD subject. So these slides explain why we select the attribute set, which contains 5 attributes. As we show in this figure, for the all subjects classification and male-female separate classification, we got best classification accuracy when we are using the combination of all 5 attribute sets for the computing of the distance of the intergraph distance compared to any other combination of those or any other subset of those attributes. So the main contribution of this work is use of entire network structure in the classification framework. Also the reduction of computation cost because we represent the network efficiently by cluster of voxels. So compared to the 28,000 voxels, our node number is in average 60 per network. Also, we got state-of-the-art detection rate, which is around 73%. So, so far we actually perform our classification only on fMRI data. In the last chapter, we try to find the answer of the following questions. First, is SMRI data is useful for ADHD classification or not? Second, if we combine this information with fMRI data information, can you further improve the classification accuracy or not? So for this work, we use multimodal data. For SMRI data, we use gray matter images. For fMRI data, we use the power map images, which is derived from fMRI data, and this power map computation approach is similar to the approach we discussed in the last chapter. So two frameworks are used for two data types. First, we use convolutional neural network to extract gray matter features, and we use local binary pattern texture feature in 3 orthogonal directions on the power map images to extract features. Then we perform the fusion of gray matter and power map image features, and we found when you are combining the information of the both modality, we are getting the best classification accuracy. So let's look at the first framework, which use convolution neural network to extract gray matter feature. So before going into the method details, I want to talk about the CNN architecture. Convolution neural network is a variation of multi-layer perceptron, which is a feed-forward network. And convolution network and CNN's architecture are motivated by the construction or organizations of neurons in visual cortex. And CNN is specifically designed for processing or classifying images. So each layer of the CNN performs two tasks, first convolution, then subsampling, and we will see shortly what those means. And the advantage of the CNN over the general neural network is, first, it can identify the spatial orientation of the input. Second, it has the property of position invariance and scale invariance. Also, the number of connection is much lower than general neural network. So here is the architecture we used. It's designed by Berkeley Vision Group, and it's called CAFE. So this is a pre-trained network which is trained on 1.2 million of the images from 1,000 different categories. So first 5 of the layers perform convolution and subsampling. So given an input image and a filter, convolution of filter, the convolution filters can perform convolution on the image to generate the convolved image, and all the convolutional filters perform the convolution on the input image to generate the feature map. And then the last 2 layers is called FC6 and FC7 layers. These are fully connected layers which are connected to all the input of the previous layers. And there is one more layer available, and that's used for classification, but we are not using it. Instead, what we are doing, we are extracting features from FC6 and FC7 layer and concatenating those to form our feature vector. So here is the whole framework of our approach. Given the gray matter images in different slices, we input those slices into CNN and extract feature from FC6 and FC7 layer to construct the feature vector for each of the layer. And from now on, we actually consider each of the slice of the gray matter image separately, and using the feature vectors of each of the gray matter layer, we are actually classifying n classifier, where n is the number of input slice of the gray matter image. So each of these classifier can independently classify a test subject and forming a decision vector. Then we can multiply this decision vector with a weight vector to come up with the final decision score. And we'll show shortly how we compute the weight vector. So here is the image for the gray matter images, which are the segmentation of the gray matter regions of the brain. Here is an example of the filters which learned by CNN in the first layer. As you can see, it can capture different orientation of the edge, texture feature, also color gradient. And here is the feature map generated by the first layer of CNN. Similarly, the example of the filters and features of the second layer. So in this slide, we actually explain how we compute the weight vector. It's a 2-step process. In the first stage, we actually compute initial weight vector. It's performed by Lee-Von-Wout cross validation on the training set of data using each of the layer. And each of the layer actually produce a classification accuracy on the training set, and those accuracy is recorded and normalized to form a weight vector omega, and this is our initial weight vector. In the second stage, we actually multiplied each of the weight vector by a function which is a inverse sigmoid function, and the idea is like giving more importance to the layers which has higher weights and lower importance to the layer which have lower weights. So this is called the rescaled weight vector, and we use this rescaled weight vector to combine the information of the different gray matter images. As I told, we use like a pre-trained model of CNN for our classification, but there are possibility to like train the CNN. There are two kind of possibility. First, we can train a CNN from the scratch, or we can get a pre-trained CNN and fine-tune them using our images. So there are challenges for both of these ways. First is the CNN requires a huge number of training samples for training the data set, training the parameter. Second, we need to decide on different number of parameters. For example, the learning rate of the network, the number of features available, number of layers available in the network, or a number of features dimension, all those things. So we did some experiment on the fine-tuning. What we did, we actually tried to fine-tune the last two layers, which are the fully connected layers of the network. And we observed like 6 to 10% improvement of the classification on the training data. And we are planning to perform more experiments to conclude these experiments. In the second architecture, we used the LBP texture feature in 3 orthogonal directions on the power map image to extract feature. So the classification framework is like this. Given a 4D fMRI data, we compute the 3D voxel power map, and again the voxel power map computation is similar to the last chapter as we described. Then we actually viewed the 3D power map image as a 2D slice in 3 orthogonal plane directions, and each of these plane direction we compute the LBP texture feature. And LBP texture feature is a 255 dimensional histogram each of the plane direction, and we concatenate the information in 3 plane direction to form our feature vector, and finally we perform classification using our classifier. So LBP feature computation step is as follows. Given a 2D image like this, for each voxels in this image, we extract the value of all neighboring voxels, and then use the middle voxels as the threshold value to binarize the neighboring voxels between 0 to 1, and then multiplies those binary vector by different power of 2's in a particular order to come up with the LBP score, which is 60 in this case. So after computation of LBP score for all voxels in each plane direction, we can compute the histogram of LBP score. So what LBP actually captures? First, if all the neighboring nodes value is lower than the center node, it can represent as a spot, and it is actually represented by a particular bin of the histogram. Similarly, it can capture different orientation of the edge, a corner, or another orientation of the edge. So it can capture altogether 255 different texture feature. In this image, we are actually showing the difference of the power map. In the top row, it's showing difference of the power map of the control and ADHD subject. In the bottom row, we are showing ADHD minus control subjects. As you can see, like in the control minus ADHD subjects, the high power regions are actually distributed evenly in the whole brain regions, while in the ADHD minus control subjects, high power regions are clustered in some regions. So these are the pattern of power map difference which helping us to classify the ADHD subject with higher accuracy. In the final phase, we actually perform the fusion of 2 different data modalities. For that, we use a voting method, and what we said is like if a subject is identified as ADHD by any of the framework, then that subject is classified as ADHD. So for the experimental setup, what we do is we use the releases for the training of the classifier, and we use the holdout set for the validation. For feature, as we said, we use gray matter feature extracted by CNN and LBP feature on the power map, and then we perform the late fusion of those features for the final classification. And for the classifier, we use SVM for gray matter feature and PCLDA for LBP features. So here are the best results using different combination of the features. For example, top is showing gray matter on FC6 layer, gray matter FC7 layer. Then WM is representing the white matter. WB is whole brain structural images. So the main point in this figure is when you are using gray matter using FC6 and FC7 layer combined with the LBB feature, we are getting the best classification accuracy. So here is the result for all different data centers. As I said, like gray matter FC6, FC7 layer getting 74.23%, LBB features getting 77%, and combined feature is getting 79.14%. And we actually beat the current state-of-the-art classification accuracy by a long margin. So the main contribution of this dissertation is, first, we efficiently model the brain function as a connectivity network using the fMRI data, and then you exploit the topological difference of the brain networks for final classification. Again, we propose an algorithm to identify the important brain regions, which shows like when you are using the features from only those regions, it improves the classification accuracy. Also, those regions are independently identified by other researchers on ADHD. We proposed the idea of power map, and we showed like using the power map can reduce the dimensions of the network to efficiently compute the network, also improve the classification accuracy. We also showed the usefulness of structural data for the ADHD classification, and we showed like fusion of structural and functional data can improve the classification again. So for the future work, we actually computed the network where network are the statics, because the weight of the network edges doesn't change over the time, because we compute the correlation of time series, the whole time series at a time. But what we can do, we can actually compute the correlation of two windows of the time series and slide those windows over the time and compute the correlation each time. That will make the edge weights a function of time again. And it will be interesting to see if the change of pattern of correlation values in the different ages can show any difference in the ADHD and control subjects or not. Also, for constructing our network in the third approach, we used a particular ROI map. It's called CC200. There are other ROI maps available, and we need to verify which ROI map is most effective. Also, we showed like the use of phenotypic information like male-female information can improve the classification accuracy. We need to validate all setups phenotypic information available and find out which are the information which are most useful for ADHD classification. Again, for the CNN, we can finish up the fine-tuning experiments to conclude what fine-tuning actually can provide. Also, we can input in the CNN the whole 3D volume of the gray matter instead of a slice by slice input, and observe like if that can improve the classification accuracy or not. So here is the related publication I have, and thank you, and I can attend or address any questions you have.